Hello students, welcome back to Facts in Biology, a YouTube channel that is devoted for the preparation of your NEET examination. Through this channel, continuously we are attempting to make you aware about the in-depth information about the syllabus of CBSE, NEET and the state board also. Friends, myself Anand Joshi, a teacher in biology. And if you find these videos helpful for your preparation, you please subscribe, like, share and strike the bell button so that very easily you can get notified whenever a new video is uploaded. So today we are going to discuss a very important concept in angiosperms, and obviously it is pollen-pistil interaction. Because we know in the last video we have discussed the process of pollination, and once the pollination is over, the next phase it is going to be the pollen-pistil interaction. Now actually the sequence of events from deposition of pollen grain onto the stigma of gynoecium to the entry of pollen tube within the ovule, the entire sequence of events, these are described as pollen-pistil interaction. Now actually it is a dynamic process in which pollen recognition followed by either promotion or inhibition of pollen germination takes place. The pistil or stigma of gynoecium has ability to recognize the pollen grain, and it involves a very complex biochemical, physiological and morphological mechanism. Friends, if a right type of pollen grain is deposited onto the stigma, the pistil accepts it and promotes it post-pollination process or fertilization. But if the wrong type of pollen grain is deposited, it will be rejected and the germination of it will be prevented. Now here in this diagram you can see a correct type of pollen grain is deposited, hence the germination process is continued. But here you can see these are the pollen grain that belongs to wrong type and their germination is prevented by some biochemical mechanism. Now actually the germination pollen grain is a dynamic process that involves biochemical, physiological and morphological events. The pollen tube which is produced after germination, it penetrates through the stigma to style and then into the ovary and finally into ovule through the small opening at the tip that is called as micropyle. Now during this process, pollen grain represent 2 or 3-celled male gametophyte, the mature male gametophyte. Now if we see the process of double fertilization and triple fusion, that is a characteristic feature of angiosperm. Actually the process of fusion of haploid male gamete with the haploid female gamete to form a diploid zygote, the process is called as fertilization. Very importantly, here you can understand that there is a fusion of haploid male gametes as well as female gamete. So both of them are haploid so that they can continue the diploidy of the sporophytic generation. Now fusion of one male gamete with egg and other male gamete with the secondary nucleus is named as double fertilization. As the fertilization occurs at 2 steps, it occurs at 2 stages, it occurs at 2 different places, the process is called as double fertilization. All angiosperm, they are characterized by the unique process of double fertilization and triple fusion. Now actually this double fertilization triple fusion process was discovered by S.G. Navashin in Lilium and Fritillaria plants. The process of double fertilization triple fusion, it is completed in different stages. Some of them they are as follows. The first, after fertilization or after pollination, pollen grains germinate onto the stigma to form a long pollen tube. Now this pollen tube, it penetrates through, it pierces through the downside into the stigma, into the style, into the ovary and finally towards the ovule due to secretion of some boron material. And this process of attraction of pollen tube towards the source of chemical, it is described as chemotaxis. Now actually this pollen tube, it carries 2 non-motile male gamete, little bit of cytoplasm and degenerating tube nucleus. The growth of the pollen tube occurs at its tip where most of the cytoplasm is concentrated. Additionally, The plant hormone auxin also plays important role in this movement. On reaching the ovary, the pollen tube, mostly it enters into the ovule through the small opening called as micropyle. And if the pollen tube enters into the ovule from the micropyle through the micropyle, the process is called as porogamy. Here in the diagram you can see the entry of pollen tube is through the micropyle, and this process is called as porogamy. But many times it is observed that the entry of pollen tube into the ovule, it occurs through the integument, and this process in which the pollen tube enters into ovule through the integument, it is called as mesogamy. Another situation is that where the entry of pollen tube into the ovule, it occurs through the chalaza from the opposite tip of the ovule. Now this process is described as chalazogamy. But irrespective of the place of entry into the ovule, the pollen tube is always attracted, enters the embryo sac near the egg apparatus only. Now we know the filiform apparatus of synergid. They attract the pollen tube towards the egg apparatus by the process of chemotaxis. Now the tip of the pollen tube absorbs water from the synergid and bursts open to release two male gametes. The tube nucleus, by this time it undergo degeneration, and the released male gamete are now free into the embryo sac and available for double fertilization and triple fusion. Now actually if we see the process of fertilization, it is completed in two stages. The stage 1 is called as syngamy, or it is called as first fertilization. Now one of the male gamete, in fact the first male gamete, it fuses with the egg or oosphere to form a diploid zygote, or it is called as oospore. Now this is termed as first fertilization, or it is called as syngamy. Mind it well, here the first male gamete, it is used, and secondly the egg or oosphere is used, which is now converted to oospore or zygote. The second process or second fertilization, it is also described as triple fusion. The other male gamete fuses with the diploid secondary nucleus to form a triploid primary endosperm nucleus, and that's why it is described as triple fertilization, triple fusion or second fertilization. Here the secondary nucleus is produced from the two polar nuclei which are coming down into the center from this we can say antipodals as well as egg apparatus. Since each of the polar nuclei is a sister nucleus of egg, it is termed as second fertilization. Now this fertilization process in which non-motile male gametes are transported to the female gamete by using a pollen tube, the process is described as siphonogamy, because siphonogamy is another term that is used for describing the pollen tube. Now if we see the fertilization process, it has got significance, and these include the double fertilization is significant because it involves use of both the male gametes. There is no wastage of male gamete. In fact, there is, we can say, a security which is we can say taken into account to avoid the chances of polyembryony and to increase the chances of survival of future plant. It consists of two fusions and results into formation of two products. The first product is obviously diploid zygote. The second one is the triploid pane. And this diploid zygote, it develops into the embryo, and in future it will give rise to the daughter plant. Another thing we can say is that second fertilization product, that is triploid primary endosperm nucleus, very soon it is going to get developed into a nutritive tissue, characteristically triploid endosperm in angiosperm. And as the development of embryo and endosperm takes place close to each other, proper nutrition of the embryo is ensured. Additionally, we can say the development of embryo and endosperm is simultaneous. Therefore, unnecessary wastage of endosperm is avoided, metabolic energy is saved by the angiospermic plant. And very importantly, it is a characteristic feature, unique feature, which ensures the parent plant invests in a seed with food store only if the egg is fertilized, so that there is the assurance of next generation and continuation of the life cycle. Friends, these are, we can say, the significance of double fertilization, triple fusion. If you find these videos helpful, please subscribe, like and share to my channel. Thank you very much.